All right, I'm going to attempt to act out theatrically here DNA replication. The enzyme helicase is going to go to a spot that's rich in A's and T's, open up DNA, separate the strands at that point. It'll be called the origin of replication. We're going to start over here on this side, helicase. All right, so there's a replication fork. Next thing, we need a primer. Primase. Primase is going to place an RNA primer down in the 5' to 3' direction, providing the all-important 3' hydroxyl group.
Go, go, go!
Start over here, 5' to 3'. Over here, 5' to 3'. Next one, polymerase 3. Polymerase 3 cannot start de novo. It can't start from nothing to attach to, so it's going to require a pre-existing 3' hydroxyl group. That's what the primer is going to supply. So here we go. On this side, here it is attaching. That's representing a covalent bond, and then polymerize, 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 polymerize. That's called the leading strand. It's going to be continuous. Meanwhile, on the lagging strand, we're going to do something similar. Attach to the 3' hydroxyl group here, which you might see there, and go in the 5' to 3' direction. And this is going to go this way. I'm going to tear that off here at this point. All right, 5' to 3 in that direction. That's called the lagging strand. Next one, primase again. Oh, sorry, helicase. So helicase continues. Helicase continues. Replication fork. Primase. Primase comes along on the lagging strand, places a primer. Next one. Polymerase 3 again. Polymerase 3 is, this polymerase 3 is on the leading strand. It's going to just keep on going. Meanwhile, on the lagging strand, we're going to attach to the 3' hydroxyl group of the RNA primer, and we're going to keep going until we get to the back end of the other primer, and we'll stop there. Helicase. Helicase again. We'll go all the way to the end with this one. We'll remove that. Make a little space here. Make a little space there so you can see it better. All right, next thing, primase. Primer comes along. Places a primer, 5' to 3' direction. Next one, polymerase 3. Polymerase 3 is going to continue on the leading strand. And we'll say that we got to the end here. That's the leading strand, continuous. Polymerase 3 on the lagging strand. Attach to the 3' hydroxyl group. Go to the back end of the other one. And you have that. Good. All right, so I'm going to slow down now and introduce you to polymerase 1. Polymerase 1. So polymerase 1 is what's called an exonuclease, which means it can cut out nucleotides, and it's also an endonuclease, which means it can put new nucleotides in. By the way, the polymerase 3 is just an endonuclease. It's not an exonuclease. This can do both. So we're going to do that. We're going to go to some of these primers. We're going to remove them, so exonuclease, and then replace them with DNA, which of course is what this stuff is here. So I'm going to start at the ends here, and I'm going to remove this primer, but I'm not going to replace it. Not going to replace this one. The reason for that is there's no 3' hydroxyl group to go 5 to 3. So that's going to shorten that half. That's part of the end replication problem. So we're just going to remove but not replace. Same thing over here. We're going to remove but not replace. So this end got a little shorter too. So that's the end replication problem. But for these internal ones, it'll work out. So we'll use this. The DNA polymerase, which way do I want? I got to go this way. And we'll remove, and replace with new DNA. Okay, next one. Remove and replace with new DNA. All right, good. So we're almost done. We're down to one more enzyme. It's called ligase, and I'm going to use a scissor for ligase. Ligase covalently bonds phosphodiester bond the adjacent nucleotides at all the places where there are little gaps. So let's go ahead and do that. So ligase, covalent bond. Ligase, covalent bond. Ligase, covalent bond. Ligase, covalent bond. Last thing will be to put down the directional numbers, 3', 5' in the right places. So 5 here, 3 there, 5 here, and 3 there. And what you have now is DNA has been copied. And that's DNA replication, but then there's the end shortening thing, which is called the end replication problem. And the solution to that will be telomerase. This is telomerase, the solution to the end replication problem. And what it is a protein that has embedded in it an RNA sequence. Please take note of the 3' and 5'. And what it does is it attaches itself to the 3' end of one of the 2 strands of DNA. And that RNA sequence here is actually complementary to this end sequence, so that's why it connects there. And then it's going to grow this portion in the 5 to 3 direction with DNA. So here it goes. 5 to 3, 5 to 3, 5 to 3, 5 to 3, telomeric DNA. So it just made this strand longer than it normally would be. Now, this helps because now that the enzyme is gone, we can do the steps of DNA replication. So that would start with the primer, primase. Remember this thing? And so the primer would be placed down right here, 5', 3' going this way. And then polymerase 3 will show up and attach to the 3' hydroxyl group and polymerize in that direction. Then, then DNA polymerase 1, which is both an exonuclease and an exonuclease, it's going to do the removal function, so exonuclease, but it cannot replace that. So this side still gets shorter in the end, but it doesn't matter because we made it longer before we made it shorter. It's sort of like 5 steps backwards, 2 steps forwards, you're still, on the balance, you didn't shorten DNA. And that is how telomerase lengthens the DNA first before it gets shortened. So in the end, it kind of stays the same. We're going to use this climbing rope to represent DNA. When replication happens, DNA has to open up. And it's a replication fork is what that's called. Now, as that replication fork travels, it could accumulate tremendous amounts of tension or helical tension ahead of that replication fork. There is a solution to that, and it comes in the form of an enzyme called gyrase, which belongs to a category of proteins called topoisomerases. I'm going to show you how that works in the next little section.
For this video, I'm going to replace the climbing rope with paper because the enzyme really can't cut through rope, but it can cut through paper. But paper now represents DNA just as the rope did. So ahead of the replication fork, tension can build up, but luckily we have an enzyme called gyrase, which has two functions. There's the cutting of DNA function, and then there's the covalently bonding those recently cut pieces together function. So ahead of the replication fork, there's tension, and the gyrase enzyme is going to cut one of the two strands. So cut, then untwist one of the two strands, and then covalently bond those two pieces back together again, like that, and you've reduced the tension ahead of the replication fork. And the enzyme gyrase does that for your cells.